Antigen-presenting cells engage in a co-stimulatory dialogue with T cells that recognize the antigens they display. This dialogue involves the delivery and receipt of signals through a number of different molecules, but appears to be initiated through the binding of B7 molecules to CD28 on a naive T cell. Antigen-presenting cells are activated to express B7 molecules on detecting the presence of infection through receptors of the innate immune system. The requirement for the simultaneous delivery of antigen-specific and co-stimulatory signals by one cell in the activation of naive T cells means that only such activated antigen-presenting cells, principally the dendritic cells that migrate into lymphoid tissue after being activated by binding and ingesting pathogens, can initiate T cell responses. This is important because not all potentially self-reactive T cells are deleted in the thymus. Peptides derived from proteins made only in specialized cells in peripheral tissues might not be encountered during negative selection of thymocytes. Self-tolerance could be broken if naive autoreactive T cells could recognize self-antigens on tissue cells and then be co-stimulated by an antigen-presenting cell, either locally or at a distant site. Thus, the requirement that the same cell presents both the specific antigen and the co-stimulatory signal is important in preventing destructive immune responses to self-tissues. Indeed, antigen binding to the T cell receptor in the absence of co-stimulation not only fails to activate the cell, it instead leads to a state called anergy, in which the T cell becomes refractory to activation by specific antigen, even when the antigen is subsequently presented to it by a professional antigen-presenting cell. Have a great day.